Hello, and thank you for listening to our video abstract on our recent work published in Current Biology, where we look at the genetic basis of spatial cognition in a food caching bird. This is a true collaboration from several universities, including the Cornell Lab of Ornithology, University of Colorado Boulder, University of Nevada, Reno, and the University of Oklahoma. I'm Carrie Branch, along with Georgi Semenov. We'll talk you through what we've found recently. So we know that spatial cognition is critical for individuals to navigate throughout their environments, and in particular for individuals that rely on seasonal migration and food storing to survive. It's especially specialized in these individuals. And we know that the hippocampus is the area of the brain responsible for organizing and processing these memories and capabilities. We study spatial cognition in this small passerine bird, the mountain chickadee. Mountain chickadees are non-migratory food caching birds that inhabit the western montane regions of North America, highlighted here in purple. And this star here represents our study site in the northeastern Sierra Nevada. And these birds rely on their specialized spatial memory abilities to store and then recover food stores throughout the winter when food is scarce. And previous work in this system has actually shown that on this rather small elevational gradient, spatial memory abilities vary with harsh winter conditions. So individuals at high elevations that experience harsher winter conditions with more snow cover and lower ambient temperatures cache more food items and have better spatial cognition compared to their lower elevation counterparts. In addition, the hippocampus is actually larger in these higher elevation birds and has more neurons compared to the low elevation birds shown here in these two figures. So while this work has shown and suggested that selection's occurring on spatial cognitive abilities of these birds, all the research to date had been indirect until our more recent work where we actually went into the wild and were able to test spatial cognition and associated fitness consequences. And the way that we've done this is we have set up multiple arrays at each of our elevations. Each one is designed with 8 feeders, and each of our birds is equipped with a passive integrative transponder tag. And this antenna here has a radio frequency identification reader inside of it, and we can actually program these feeders to only provide food to a particular individual. So when the bird lands on the feeder, if it is its correct feeder, they will receive 1 sunflower seed. So here's a closer look at that. So you can see the bird's passive integrative transponder tag on its leg, which provides the RFID reader with information on who that individual is. Using this spatial learning and memory task, we've actually shown that individuals that perform better on the task are more likely to survive the harsh winters and also produce larger broods compared to the individuals that perform worse on the task or make more errors. When we went to choose individuals for this study in particular, we chose individuals that performed the best on the task and the worst on the task across high and low elevations. While our previous work has focused on differences in high and low elevation birds, we wanted to avoid any bias that might create. So we actually chose individuals that performed the best and then the worst on this task. But as you can see here, showing the individuals actually chosen for the study from that distribution, this creates a rather continuous gradient of cognitive performance on our spatial learning and memory task. And with that, I'd like to hand it over to my co-author, Georgi Semenov, who will discuss the genomic methods and findings.
For this study, we selected 42 individuals that were more or less equally distributed between best and worst performing individuals. We sequenced whole genomes of all those individuals, and we aligned them to a reference genome of a closely related species, the black-capped chickadee. And this together resulted in over 10 million of single nucleotide polymorphisms after all the filtering steps. Next, we did genome-wide association study using 2 methods. One, a more traditional approach called GEMMA, and the other one is a novel random forest approach which is a type of machine learning. We found over 1,000 of strongly significant associations using both methods. But what was exciting is that despite very different underlying algorithm and assumptions of those two methods, over 30% of genes we found were overlapping between GEMMA and random forest, which was reassuring and suggested that we really pick up the meaningful signal of genotype-phenotype association, and also those about 300 genes that we found by both method might be the most likely candidates for determining differences in cognitive phenotype. Those genes that we found strongly associated, they are significantly represented in a number of biological pathways and gene ontology categories, and they're broadly associated with development of nervous system, and particularly with development of head and brain and hippocampus, as well as some proximate mechanisms of neurogenesis and neuron development. And of those several hundred genes, we looked closely at a small subset of genes most strongly associated with cognitive phenotype. And we found a series of very interesting genes with known function in nervous system development, and also various kinds of mental disorders. And one interesting finding for those highly associated genes, we observed very strong relationship between phenotype and the genotype for this loci, which you can see in this panel on the right, which suggests that they might be actually functionally relevant to differences in cognitive function. So broadly, the findings of our study suggest that spatial cognition in chickadees has a genetic basis and not is just based on learning and experience, as was previously suggested, which means that this spatial cognition can be actually a target of natural selection. And also we came up again with this short list of genes that might be particularly interesting for future research on brain development and cognition. And with that, we would like to thank our funding sources and all the colleagues that contributed to this study. While findings of our study are very exciting, we use a relatively small data set of 42 individuals, and in the future studies, we're going to expand to a much larger data set and also include data from parents and offsprings to see how selection shapes genetic variation of those loci, and perhaps find the genes that play the primary role. Thank you.